This is Group 18's learning resource on the comparative functions of the smooth and rough endoplasmic reticulum. So part of our learning resource is a recorded video. This isn't just a straightforward video. There are hyperlinks throughout to link you to specific sections. So for example, if you wanted to learn more about smooth endoplasmic reticulum diseases, click on the link found on this page to get you to the section specifically for the smooth endoplasmic reticulum, and then another hyperlink will take you to specifically the diseases. There has also been a handout provided along with this, for those of you that find it easier to learn by reading. And we've also made a quiz for those of you that find it easier to learn by doing. The endoplasmic reticulum is the largest organelle found in almost all eukaryotic cells. This is all except the mammalian red blood cells, and is absent in all prokaryotes. It is one continuous membrane that consists of a network of tubules, vesicles, and cisternae, which extend throughout the cytosol. The endoplasmic reticulum consists of two distinct regions, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough ER consists of flattened sacs called cisternae that connect to the nuclear membrane and are studded with membrane-bound ribosomes. However, the tubules of the smooth ER are covered in slippery fats. These associations aid their unique functions, which will be explained later on.
First, we're going to take a closer look at the rough endoplasmic reticulum. If you would like to skip to either structure, function, or diseases of the rough ER, you can click on the hyperlinks at the bottom of the page now. The rough endoplasmic reticulum is the site of synthesis of essential proteins of the body, and so is seen in almost all eukaryotic cells. The membrane is continuous with the outer membrane of the nuclear envelope, which encloses the internal cisternal space or lumen. Large amounts of rough ER are prevalent in cells that need to secrete large amounts of protein, such as plasma cells, which produce and secrete large quantities of antibodies, pancreatic acinar cells, which undergo large production and secretion of insulin, and fibroblasts and osteoblasts, which secrete organic matrix of connective tissue and bone. The rough ER is comprised of a network of flattened sacs or cisternae, surrounded by a single membrane that is continuous with the outer nuclear membrane. Despite being attached to the nuclear envelope, the ER is a highly dynamic organelle. It is able to undergo division during mitosis and is attached to the cytoskeleton, whilst maintaining its unique structure. There are many proteins within the rough ER that are also found in the smooth ER. However, there are also specific proteins localized nearer to the nuclear envelope. These proteins aid translocation of nascent proteins from ribosomes into the lumen of the ER, as well as others that are involved in processes such as glycosylation and regulation of the folding of proteins. Approximately 1/3 of all proteins encoded by the mammalian genome are synthesized at the rough endoplasmic reticulum. These proteins include secretory proteins, lysosomal proteins, and integral membrane proteins, which reside in both the membranes of cells and the membranes of other organelles. The first process of the rough ER is translocation. This is the movement of a nascent protein on a membrane-bound ribosome into the ER lumen. It is also a highly conserved mechanism. A polypeptide that is being translated on a ribosome contains a hydrophobic signal sequence at its N-terminal. This signal sequence is recognized by and binds to a signal recognition particle, or SRP. The signal recognition particle has the ability to temporarily reduce synthesis of the nascent protein on a ribosome. This delay in translation is important to allow for co-translational translocation of the polypeptide into the ER lumen. If it is not delayed, it may lead to secretion problems later on and may slow down cell growth. There is a signal recognition protein receptor localized on the membrane of the ER. The SRP binds to this receptor, therefore bringing the polypeptide ribosome complex to the ER membrane, where the ribosome is held in place by ribofourin proteins that attach to the large subunits of the ribosome. The binding of the SRP with its receptor is regulated by a GTPase cycle. Once bound, hydrolysis of a GTP molecule on the SRP will cause the signal sequence to dissociate, allowing the polypeptide to enter a translocon and allowing translation to resume. The translocon is a protein channel embedded in the ER membrane with a hydrophobic poring through which the protein moves to the ER lumen. Once the polypeptide has been translated, the membrane-bound ribosome detaches, the polypeptide moves into the lumen of the ER, and translocation is complete. Another function of the rough ER is the glycosylation of proteins. Most proteins synthesized in the rough ER are glycosylated by the addition of an N-linked oligosaccharide. And approximately 50% of proteins processed by the endoplasmic reticulum are glycoproteins that are modified in this way. The most common form of protein glycosylation in the ER is where a 14 sugar molecule called a precursor polysaccharide is transferred onto the protein. The precursor polysaccharide consists of N-acetylglucosamine, mannose, and glucose. Calnexin and calreticulin are ER chaperone proteins that only recognize N-linked oligosaccharides that have a single terminal glucose. This means that the chaperone proteins only recognize glycoproteins that have undergone a trimming process, in which 2 glucose molecules are removed from the precursor N-linked oligosaccharide. When the third and final glucose is removed, the glycoprotein can dissociate from the chaperone proteins and leave the ER. Glycosylation in the ER also monitors protein folding. An enzyme called glycosyltransferase adds a glucose to the oligosaccharide chains that have lost their terminal glucose, but only to those proteins that are incompletely folded. Proteins that are not yet completely folded undergo a cycle of having their terminal glucose trimmed and then added again until the protein is completely folded, at which point it can leave the ER. Most proteins will then travel to the Golgi. Receptors in the trans-Golgi network only bind to specific sugar moieties, ensuring that proteins reach their correct destination. Glycosylation is vital for normal protein function in mammals. Therefore, proteins that are required for human treatment cannot be raised in prokaryotes. This is due to the lack of an endoplasmic reticulum, or Golgi, and any N-linked or O-linked glycosylation respectively. To mass-produce human proteins that have undergone this form of post-translational modification, transgenic animals are used. That is, if fungal post-translational modification is not specific enough. One such example of a transgenic animal is the Atrin goat, which produces a human anticoagulant in their milk. Issues with function in the rough ER are seen in many prevalent diseases, such as vascular and cardiac diseases, cystic fibrosis, diabetic kidney disease, Parkinson's disease, and many more. The functional issue most commonly found as a cause of disease is endoplasmic reticulum stress. This is when there is a problem in the folding of proteins produced in the rough ER. As a result, the proteins are misfolded or not folded at all. These incorrectly folded proteins accumulate in the lumen of the endoplasmic reticulum and initiate a response called the unfolded protein response, or UPR. This response attempts to alleviate the ER stress and restore homeostasis, but in cases where that is not possible, it will cause apoptosis of the cell. If the glycosylation function of the ER is impaired by a genetic mutation, it will cause disorders that affect almost all organs due to the inherent nature of the endoplasmic reticulum. These disorders are called congenital disorders of glycosylation, CDGs. There are 40 defects associated with N-linked glycosylation, and most cause recognizable syndromes, such as phosphomonomutase 2, or PMM2 CDG, phosphomannose isomerase, or MPI CDG, ALG11 CDG, and RTF1 CDG.
Now let's take a look at the smooth endoplasmic reticulum. If you would like to skip to the structure, function, or diseases of the smooth ER, you can click on the hyperlinks now. The ribosome-free part of the endoplasmic reticulum is known as the smooth endoplasmic reticulum, the SER. As it has no ribosomes, it has nothing to do with the synthesis of proteins. It is the synthesis of lipids and steroids that the smooth ER has an important role in. There are ER exit sites on the SER that make it possible for synthesized proteins and lipids to be transported in vesicles to the Golgi apparatus. Every cell contains smooth ER, but it is more abundant in some cells, such as the testes, the ovaries, and the hepatocytes, as they produce steroid hormones. The image shows an electron micrograph of the smooth ER. Notice the tubular structure and the absence of ribosomes. The dark spots that you can see are glycogen granules. The smooth ER is made up of tubular membranes and slippery lipids. The absence of ribosomes is advantageous as this allows easy access for the enzymes, vesicle budding, and fusion components to the lipid surface. The membrane is highly curved due to smooth ER-specific proteins, the reticulons. These proteins can bend the membrane to form the tubule. Other proteins in the membrane, flipases, cause phospholipid flip-flop. The reticulons are mainly seen in the smooth ER but can also be seen throughout the rough ER as it is involved in endoplasmic reticulum Golgi trafficking, the formation of vesicles and membrane morphogenesis. Curving of the membrane happens when the reticulons interact with and ER integral membrane protein DP1 YOP1P. Although the SER has no role in protein synthesis, it has plenty of other functions. One function includes drug detoxification. Cytochrome P450 enzymes are found in the smooth ER and their function is to hydroxylate hydrophobic carcinogens and other compounds. They are usually converted to water-soluble compounds and are excreted out of the body. This happens in the liver cells, the hepatocytes, which explains why there is more smooth ER present in these cells compared to other cells. The enzymes involved are often monooxygenases and aryl hydrocarbon hydroxylase. Monooxygenases work by donating 2 electrons and 2 protons to oxygen to make H2O. The other oxygen is incorporated into the drug. Aryl hydrocarbon hydroxylase is involved in converting the toxic product formed by hydroxylating polycyclic hydrocarbons in water-soluble compounds into a chemical active form. The SER is involved with carbohydrate metabolism. Glucose 6-phosphatase is an ER membrane-bound enzyme which converts glycogen into glucose between meals during exercise. Glycogen needs to be converted before leaving the cell as it is impermeable in the membrane. GLUT2 transports the glucose into the appropriate cells then. Steroid biosynthesis happens in the SER. The smooth ER is abundant in adrenal glands, the testes Leydig cells, and the follicular cells of the ovaries, as the synthesis of cholesterol, cortisol, testosterone, and estrogen occurs at the smooth ER. The electron micrograph on the right shows the SER of the testes in a guinea pig. Notice how abundant the SER is here, as the Leydig cells need the SER to produce testosterone. The smooth ER contains the enzyme hydroxymethylglutarol CoA reductase, which is a key to the synthesis of cholesterol. Monooxygenases, mentioned earlier, also have a role in converting cholesterol into the steroid hormones. Although not an independent process, the SER plays a major role in phospholipid biosynthesis along with the Golgi and mitochondria. The bulk components of the products are glycerophospholipids and sphingolipids, both of which are abundant in the phospholipid membranes. The SER is a central site for membrane biogenesis. The specific site for synthesis is known as the endomembrane compartment, which is highly involved with the Golgi. Acetyltransferase enzymes use the substrate fatty acyl-CoA to synthesize glycerolipids and sphingolipids through various steps of phosphorylation and association. Glycerolipids are also synthesized in the SER using enzymes called plasmalogens, which are produced and transferred from perioxomes. The sarcoplasmic reticulum, also known as the SR, is a specialized type of smooth ER present in muscle cells. The SR plays a big role in the storage and release of calcium ions, which aid the activation of contraction in striated muscles. The SR is specialized. The lumen has a high concentration of calcium-binding proteins. ATP-dependent calcium ATPase pump the calcium ions into the ER and release them when the muscle needs to contract. There are two forms of sarcoplasmic reticulum, the peripheral form and the central form. The peripheral form lies close to the plasma membrane of the cell and is functionally responsible for the calcium ion homeostasis of the cell, local calcium ion release, and interacting with the ion channels of the plasma membrane. The central form is more directly involved in calcium ion release to myofilaments for muscle contraction by ECC, excitation, coupling contractions. Mitochondria is in close proximity to the SR, as small as 10 nanometers away, and sometimes even engulfed by it, noticed in smooth muscle cells. Uptake of calcium ions from the mitochondria promotes maximum calcium ion release from the SR to activate muscle contractions. The sarcoplasmic reticulum is the main source of calcium ions for skeletal and smooth muscle, not for cardiac muscle, and is also more organized in striated muscles compared to smooth muscles. The calcium ion stored in the sarcoplasmic reticulum is uptaken into the SR via the SRCA ATPase, the SERCA pump. The SR volume in smooth muscle is less than the volume present in striated muscles. This suggests that the SR have different functions in smooth and striated muscle. This is probably due to the fact that smooth muscle need to contract less than striated muscles, therefore less calcium ions are required. Evidence also suggests that there is more peripheral SR in phasic muscles compared to mesenteric arteries. Calcium ions are released from smooth muscle mostly by inositol 1,4,5 triphosphate INSP3 receptor in the sarcoplasmic reticulum. The SR of striated muscles is closely associated around the transverse tubules of the muscles. This makes it easier for the calcium ions to affect the cell. Calcium ions are released from the SR by ryanodine receptors. Contraction of the cardiac muscle is mostly controlled by the release of calcium ions from the SR. The calcium ions are released from the SR through ryanodine receptors RYR2 pathways, which is affected by intracellular calcium ion concentration and phosphorylation of the receptor. The ryanodine receptor pathway is also involved in the release of calcium ions in smooth muscle, but it is the RYR1 isoform that is present in the skeletal muscle SR. The image to the right shows a sarcoplasmic reticulum of skeletal muscles. Here, the SR is blue. The T tubules are shown in yellow, and if you look closely, the SR becomes enlarged near these T tubules, and these areas are called the terminal cisternae. There are many different disorders associated with the SCR, so we have elected a few prevalent diseases where the cause and effects are well researched. Neuropathic pain is a neurological disorder in which pain arises due to the nerve injury. This occurs when the soma, or the cell body of a sensory neuron, is affected by injury to the peripheral axons. This results in depletion of the ER calcium concentration and the gene SERCA. Pancreatic beta cell diabetes results from a gradual reduction in cell mass, finally leading to cell death and consequently loss of ER. With regard to type 1 diabetes, there is a non-autoimmune disorder called Wolfram syndrome, which is caused by a mutation in the gene encoding the ER glycoprotein wolframin. The defect reduced calcium levels in the ER of beta cells and triggers the apoptic pathway. Atherosclerosis is just one example of a cardiovascular disorder of the SCR in which macrophages cause an accumulation of unesterified cholesterol, therefore changing the fluidity of the ER membrane. This can lead to excessive apoptosis and increase the progression of atherosclerosis. Downregulation of the SERCA2 gene is normal in the cells lining the airways. Cytosolic calcium levels in the cells of the smooth muscle increases and cell motility increases. Ormodal-3 is a gene encoding for an ER protein that binds to and inhibits SERCA, resulting in decreased calcium levels. Brody disease is also a mutation in SERCA1 caused by prolonged calcium elevation in the cytosol. This causes muscle cramping and difficulty to relax during exercise. This gene mutation does not lead to cell death. Next, we will look at diseases common in both the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Endoplasmic reticulum storage diseases, or ERSD, are a family of diseases that affect both the rough and the smooth ER. It mainly occurs in the parenchymal tissues of the liver and is a congenital disorder affecting the secretory proteins, resulting in hepatocytic storage. This leads to plasma deficiency of the corresponding protein due to molecular abnormality that hinders the translocation of the plasma deficient protein from the RER to the SCR. The molecular abnormality is hereditary, congenital, familial, or permanent. However, the storage disease is exclusive for the mutant protein and is predisposition for the development of chronic cryptogenic liver disease. Examples of ERSD include alpha-1 antitrispin deficiency, fibrinogen storage, or alpha-1 anti-chymotrypsin deficiency. The diagnosis of endoplasmic reticulum storage diseases involve the immunohistochemistry and electron microscopy.
On the next slide, we'll briefly summarize the rough and smooth endoplasmic reticulum. Here is the summary table comparing the structure, associated organelles, and diseases of the rough and smooth endoplasmic reticulum. So, the rough ER has a single membrane, and the membrane of the smooth ER is tubular. The rough ER has cisternae and membrane-bound ribosomes, neither of which are present at the smooth ER. Whereas the smooth ER has slippery fats, and the rough ER doesn't. The membrane of the rough ER is continuous with the outer nuclear membrane, and the smooth ER is associated with the Golgi apparatus. The two endoplasmic reticulum are also obviously associated with each other. Defects in functions of both the rough ER and smooth ER can lead to diseases in the vascular and cardiac systems. Other diseases associated with the rough ER include cystic fibrosis, diabetic kidney diseases, and Parkinson's disease. Diseases associated with the smooth ER include diabetic cardiomyopathy, diabetic neuropathy, and neurological diseases. Here's a list of references so that you can read up on any of the papers used.